Cells are constantly battling to maintain functionality, especially under stress. At the heart of this process are proteins like the ATP-powered ClpB, a hexameric molecular chaperone that releases other proteins from toxic aggregation by translocating them through its central pore. In our research, we use single-molecule fluorescence resonance energy transfer, FRET, to follow ClpB's operation. A green donor dye is attached to ClpB, while a red acceptor dye labels the tangled protein substrate. Energy transfer from the donor to the acceptor is strongly dependent on distance, making FRET a spectroscopic ruler. Initially, the donor dye emits bright green fluorescence, while the acceptor dye doesn't glow. When the substrate engages with ClpB, ATP hydrolysis pulls the substrate into the pore. The tangled protein unfolds as it enters, straightening into a linear chain. As the substrate enters ClpB and the dyes approach each other, red fluorescence increases, while green fluorescence reduces. This rise in FRET is maximal when the red dye is closest to the green dye inside the channel. As the substrate continues translocating and exits the pore, the dyes separate, FRET efficiency decreases, and green fluorescence returns. The protein exits in an unfolded state and can then refold into its functional conformation. ClpB thus restores misfolded proteins to functionality, safeguarding cellular homeostasis.